Good afternoon. My name's Sam, for those of you who don't know me. I work in the laboratory of Wayne Nicholson at the Kennedy Space Center in Florida. And my project primarily concerns the ability of a terrestrial microorganism to grow in an extraterrestrial environment, specifically a low-pressure extraterrestrial environment. So my lab, like many of your labs, is primarily an astrobiology lab. But outside of astrobiology, low-pressure adaptation in microorganisms has relevance to the field of hyperbaric food and plant preservation, as well as aeromicrobiology. It also adds to the general knowledge of extreme pressure adaptation in microorganisms. And currently, this is only known for organisms that live in the deep sea or the piezo-philes of the piezosphere. Now, to give you a reference of the low pressure, I'm going to be talking about 50 millibars. It is approximately 1/20th of the pressure that is at sea level on Earth. It is 5 times the highest pressure that is found on the Martian surface. It is also 1/2 of the lowest pressure that is found in the troposphere, and the troposphere is where all of the biosphere is currently found on Earth. So why do I care about hyperbaric adaptations in Bacillus subtilis? Well, Bacillus subtilis is a very well-known species. It is probably behind E. coli the most well-known species on this planet. Its genome is completely sequenced, and it is genetically trackable, which means it is very easy to manipulate in the lab. Now, members of the genus of Bacillus are spore formers, and spores are extremely resistant to a wide number of environmental insults, including long-term duration at space vacuum. Spores of Bacillus are also common spacecraft assembly facility contaminants. So with the continued robotic missions being sent to Mars and hopefully other extraterrestrial environments, this raises the potential threat that one of these missions could carry with it a terrestrial organism, which could subsequently contaminate the extraterrestrial environment, and this is termed forward contamination. So in 2006, Dr. Andrew Sugar, another University of Florida professor at the Kennedy Space Center, and Wayne looked at the lower pressure limit on growth of a number of Bacillus species whose spores are known to contaminate spacecraft assembly facilities. And what they found is when you get down to 25 millibars, this is inhibitory to their growth. Important to note, it is not lethal to their growth. When you bring them back up to a pressure at which they can grow, they do grow. So there's something about low pressure that is inhibitory to the cellular functions in Bacillus. Now, Wayne asked, could you adapt Bacillus subtilis to grow better at low pressure, and by doing so, study low pressure adaptation and how low pressure affects cells? So along these lines, he set up a 1,000-generation continuous culture experiment at 50 millibars, 27 degrees C, and LB media. The input, or the ancestral strain, is our laboratory 624. Now, 624 doesn't grow very well at 50 millibars. It reaches an optical density of approximately 40 clet units. During the 1,000-generation experiment, weekly average optical densities seemed to indicate periods of punctuated evolution. At the terminus of the experiment, a strain was isolated from the growth media, which we call 1106. Now, 1106 grows to a higher optical density at 50 millibars, reaching approximately 80 to 85 clet units. It also has a higher relative fitness than the ancestor, meaning that if you compete them in a flask at 50 millibars, the ancestor gets knocked out of the growth media. So the first series of experiments looking at the differences between hypobaric evolved 1106 and the ancestral 624 was a series of transcriptional microarrays. Now, there were some similarities in their response to low pressure. They're both still very stressed out at low pressure. So they turn on their general stress response, which is dependent upon the alternative transcription factor sigma B. They also, due to lowering the pressure in the headspace above the media, you're lowering the oxygen available for the bugs. So they both turn on their hypoxic-related signals. Now, there's some dissimilarities at low pressure. In the hyperbaric evolved strain, it upregulates a cis operon, which is involved in sulfate metabolism. There's also a difference in their iron responses at low pressure. And in the ancestral 624 strain, it highly induces its flagella operon, and this isn't seen in 1106. Now, across all of my pressure transcriptional microarrays, there's approximately 1,000 genes that are differentially expressed. That is about 25% of Bacillus subtilis' genome, and a lot of hay to sort through for a couple needles for my dissertation. So, as I said, during the 1,000 generations, it looks like there are periods of punctuated evolutionary changes. That leads one to believe that there were specific changes to the genome that gave rise to the phenotype that we see in 1106 at 50 millibars. So I looked for these changes by conducting whole genome resequencing, And so far in my analysis, I have found 10 single nucleotide polymorphisms, or SNPs. Now, what's interesting is 4 of these occurred in a 10 nucleotide stretch of the RNJB coding region. Now, when I PCR amplified and resequenced this region, I found that these are not SNPs at all, but in fact a 9 nucleotide deletion, which results in a 3 codon in-frame shift. So what you're seeing here is the region of interest in the ancestral strain, and then below that is my hyperbaric evolved protein sequence. And it resulted in the deletion of an alanine, a lysine, and an isoleucine. Now, RNJB codes for RNase J2. RNase Js are members of the beta-Cas family, which is within the superfamily of the metallo-beta-lactamase family. Members of this family work on either RNA or DNA substrates. RNase Js work on RNA substrates. So in Bacillus subtilis, RNase J2 is involved in global messenger RNA processing in the cell. It is an endonuclease with extremely low 5' to 3' exonuclease activity. In the cell, it forms heterodimers with RNase J1, and it's believed that the predominant form of the RNase J1, J2 complex is actually a tetramer. Now, RNase J1 is essential. It has the main 5' to 3' exonuclease activity in the complex, and it is also an endonuclease activity, has endonuclease activity. RNase J2 is non-essential. However, when RNase J2 binds with RNase J1, it affects the substrate specificity of the endonuclease activity of RNase J1. So what are the possible effects of this mutation on my mutant protein? Well, the 3 codon deletion happened in a very highly conserved region of RNase Js, which forms the helix 5 structure. So I thought, okay, if you delete some amino acids in a helix, you're possibly disrupting the rest of the helix. So I did an in silico analysis for secondary structure prediction. And what you're seeing here is the top sequence are the amino acids in the wild type protein that form helix. Underneath that is my mutant, the corresponding amino acids in my mutant, and they no longer have helix hits. Underneath that is the corresponding amino acids that form helix 5 in RNase J1. Now, RNase J1 of Bacillus subtilis has recently been crystallized. So I did a tertiary structure prediction, which agrees with the secondary structure prediction. On your left is the mutant protein, and as you can see, that helix 5 is no longer forming. This is the corresponding helix 5 in RNase J1. Now, this is believed to be how the RNase J1, J2 complex looks like in the cell. I've highlighted the helix 5 in pink, and as you can see, it's on the tetramer interface. It also occurs in a stretch of amino acids that separates the metallo-beta-lactamase domain from the beta-Cas domain. And this is unique to the beta-Cas subfamily, and it ties the 2 domains together and could possibly have impacts on their catalytic efficiency. So the mutations in 1106's RNJB may be causing structural changes that affect the messenger RNA processing of this RNA J1, J2 complex. This in turn may lead to an increase in messenger RNA half-life, which at 50 millibars may cause an increase in product. There may also yet be an unknown specific activity of RNase J2, and this activity may be disrupted by these mutations, and the disruption of this activity could be affecting the essential function of RNase J1. So to test these hypotheses, I recently received an RNJB mutant from Dr. Stolke's lab, a very generous gift, and I'm using this to transform my hyperbaric evolved 1106 in my ancestral strain, and then I'm going to compete the mutants and see what their fitness is at 50 millibars. I'd also like to do a messenger RNA decay rate to see if any of the substrates of the RNase J1, J2 complex have an increase in half-life or possibly a decrease in half-life. And I would also like to do a protein analysis of the RNA J1, J2 complex in 1106 to see if the tetramer is being disrupted and possibly maybe even the heterodimer as well. But ultimately, I would like to tie in these mutations to what we saw in the continuous culture experiment. So I would like to test the population before an optical density jump and after an optical density jump. And I could see one of 2 things. I could see that this 9 nucleotide deletion happened all at once, or it slowly happened, and there were multiple little deletion events that occurred and ultimately gave rise to the deletion of these 3 amino acids. Thank you.
So any questions for Samantha?
Hey, I was just wondering, why is low pressure bad for bacteria? Why don't they like that? Why is it inhibitory?
So many reasons. Well, so pressure is sometimes compared to temperature, where high pressure is compared to cold temperature, extreme cold temperature. Low pressure could be compared to high temperature, so you could be having too much expansion of catalytic, like enzymes. The membrane could become too fluid, so you could have leakage across the membrane of water molecules and other things. But honestly, nobody knows because nobody studies low pressure at all, except for apparently me. So the only thing that I've seen is they'll take plants down to 100 millibars, which is still, not as low as bacteria can go, obviously, down to 25 millibars, or even lower in some cases to Martian pressure, 7 millibars. But it could be. So many things go wrong, and including in eukaryotes, there's an increase in oxidative stress too. You have an increase of reactive oxygen and nitrogen species, RONs, when they expose mice and other animals to like low pressure. So lots of things.
Is it a homogeneous population at the end, or have you seen any distinct different populations within the continuous growth culture?
For the continuous culture, so one strain was isolated from the growth culture. That doesn't mean that there aren't more hyperbaric evolved strains in there, but right now I just study 1106. Okay. So yes, looking at the whole population is actually something my boss wants to do at some point. Yeah.
Do the strains that adapt to high pressure still function well on low pressure, their original condition, or is there an evolutionary trade-off?
Do I need to repeat that question?
If you can.
So do strains that have been adapted to high pressure still grow at low pressure?
Yes.
Okay. So I have not adapted a strain to high pressure and grown it at low pressure. However, there is a genus of bacteria, the first gram-positive that was isolated from the piezosphere is Carnobacterium. Carnobacterium also grows at Martian conditions, so extremely low pressure. So it is a very plastic genus. So it is possible for an organism to grow both at high pressure and low pressure. I hope I answered that.
So thank you for your talk. That was great. Oh.